Well, good afternoon, everybody. I'm Debbie Ingalls. I'm the director of CCOVI here at Brock University, and welcome to the continuation of our 2024 lecture series. It's my pleasure to introduce our speaker today, Dr. Malkie Spodek, who is CCOVI Scientist in Entomology and an adjunct professor in the Department of Biological Sciences here at Brock University. Malkie is an entomologist with recognized expertise in systematics, biosecurity, biological control, and integrated pest management in agricultural systems. She earned her master's and PhD in agroecology and plant health at the Hebrew University of Jerusalem in Israel. Working as a research entomologist in Israel, she investigated local natural insect enemies to control cotton mealybug in sweet pepper net houses and examined insect fauna diversity feeding on a damaging weed in date plantations. Malkie is a hemipteran taxonomist, that's a mouthful, having found and described several new species of insects during the course of her work. You might know them more commonly as everything from bedbugs to aphids to cicadas or assassin bugs. You have to tell me about those. I don't know what those are. At CCOVI, Malkie works with the $5.5 billion Ontario grape and wine industry to understand the needs for insect pest management, with several projects under review for funding. She's a member of the Entomological Societies of Ontario and of Canada, and sits on several advisory committees, such as the Fruit Technical Working Group of Ontario and the Spotted Lanternfly Research Coordination Group. So please join me in welcoming Dr. Spodek and her presentation today on the laboratory trials of entomopathogens as biocontrol treatments for grape mealybug.
Thanks, Debbie, for that introduction. Good afternoon to you all. Wow, it's nice to hear my voice. Everybody can hear, including me back there. Okay, I'm excited to be here and to deliver my very first talk in the CCOVI Lecture Series. Today I'm going to talk about a project that involves several partners, as you can see here. Some of them are in the room today. Actually, 3 out of 4 are in the room today. One of the partners is Dr. Sid Pujari, who is a plant virologist with CCOVI and adjunct at Brock University. He introduced me to Ontario mealybugs last summer in 2023 when I first started, and actually inspired the subject for this talk today. Dr. Wendy McFadden-Smith probably needs no introduction, but I will introduce her nonetheless as IPM specialist at OMAFRA, and she's been working with grape mealybugs for some years now and shared her wealth and knowledge and experience and helped me find them in local vineyards. Essential partner to this project despite being a phytopathologist by training. And Dr. Badra Vamalpati is a he's a research postdoctoral fellowship and has been working with Dr. Pujari at CCOVI for the past year. And his experience and expertise with insect pathology has been a great asset to the team. And last but not certainly least, I'd like to introduce Olivia DeVries, who is sitting here today. She's a third-year biotechnology student at Brock University, undergrad, and she was hired as my research assistant. I also call her the mealybug whisperer these days, because she was also a very essential part of getting this research program off the ground. The funders, I would like to thank OGWRI and Greenhouse Technology Network, and Explore Grant from Brock University are the main funders. Today, this is the outline that I'm going to be talking about. I'll introduce a little bit about mealybugs, their importance in the wine industry, going to shed some new light with some diagnostic information about the grape mealybug in Ontario, discovering its identity, current management and treatment of the grape mealybug in Ontario, the successes and challenges of establishing a laboratory rearing of this mealybug in order to then perform laboratory trials using this entomopathogenic fungi, which I will go into much more details later, and of course end with some future research directions. So, 2 weeks back, we heard from Dr. Sud Pujari about the effects of viruses on vines and the effect that it has on Ontario's grape and wine industry. Now, the industry faces devastating economic and sustainability challenges from these plant viral diseases, and they're spreading at unprecedented rates. So the primary spread of propagating virus-infected planting material is from infected mother vines. And these here are some of the main viruses that we see: grapevine leaf roll virus, grapevine red blotch disease, grapevine family virus, and Pinot Gris virus. So new technologies are being developed at CCOVI to eliminate the primary source of virus-infected vines, including clonal propagation of infected mother vines to generate planting material. This clean plant material, coupled with green grafting technology, is currently under development. And it will allow nurseries to join the clean rootstock and clean scion material to produce a new generation of grafted vines. However, insect vectors are also responsible for the secondary spread in vineyards. And here is a list of most of the culprits between soft scales and mealybugs, who are known vectors of leaf roll viruses 1, 3, and 4. These insects are also not only are virus vectors, but they also cause secondary damage to the fruit by promoting fungal infections through secretion of their honeydew. So grape mealybug and European fruit licanian scales are the 2 species that are reported here in Canada, and the remainder of my talk today will be about the grape mealybug, Pseudococcus maritimus. So some of the virus symptoms that show up on the leaves in the vineyards, it depends on the cultivar. The red and the purple coloration of the mature leaves in the red fruited cultivars seen here. And in the Merlot, we have also the back row rolling of the mature leaf margins that are commonly found in actually both red and white fruited cultivars. Uneven ripening of berries and thin clusters is also another symptom that's associated with the viruses, and this ultimately leads to reduced yield and then the poor quality of grapes. Now, the long-term productivity loss has been estimated up to close to 300,000 Canadian dollars per hectare. So this is a very serious issue that needs attention for the grape and wine industry in Ontario and Canada. But mealybugs, in case anybody wanted to know what one looks like, is here circled in red. And they are major agricultural and forestry pests. They belong to this wider order of insects known as hemipterans. Why are all these insects, most of these insects, considered pests is because of their shared feeding mouth parts. That they're able to vector viruses. And I've put these pictures of the spotted lantefly down below. Now, this is an insect that also belongs to the hemipteran order. It is not known as a virus vector, and it is not presently in Canada. But we are concerned about it as in the grape industry because of its ferocious feeding habits. So these specialized mouth parts, I don't know if anybody's ever seen a mealybug mouth part before, but these are electron scanning microscope slides, and this is actually not the grape mealybug, but a close relative. And the mouth part here is on the underside of its body. Very small insects, like about about a centimeter in length. And what you can see down here is this mouth part that's the clipulabial shield. It's triangular shaped. And this thin hair-like stylet is what actually punctures the plant tissue and acts like a straw to suck up the plant's sap, the phloem. And this is a zoomed-in image of the tip of this hair-like mouth part. So the mode of transmission of the virus is actually, for the mealybug, it's through semi-persistent, non-circulating transmission. That means the insect, here's the whitefly, but imagine it's a mealybug, will sit on the plant tissue, insert its mouth part, and then pick up the virus that's in the plant. And this cool picture here is a cross-section of a grape petiole showing the actual pathway of the stylet of the mealybug in pink here as it's inserted into the plant. So once the insect finishes feeding, the virus persists on its mouth part, and then when it moves to another plant, it will then reinfect the other plant. There's other modes of transmission for viruses, what's called persistent and circulative, where the virus can be in the insect's body, multiply, and circulate that way. So for mealybug feeding in grapevines is actually one of the most inefficient way of transmitting viruses, but nonetheless still an issue. So this here is a female mealybug and a male. They look drastically different, and one would not even think that they are the same species, but indeed they are. The males do not have mouth parts, and they are not responsible for any of the feeding or any of the virus transmissions. They do have wings, which comes in very handy, which I will explain later. And the female here is they're soft-bodied insects. They feed on vine sap, like I've mentioned, the phloem, but not on the grapes, and they spend most of their lives under the bark of the vines. And they're not known to feed on any other plants in Canada, which is important to note, and also adds to the challenges of rearing them in laboratories. But then they, like I mentioned, they're instars of vectors of vine viruses, leaf roll 1 and 3. So here we have images of a mealybug life cycle, and I'm going to draw your attention here to the egg sac. Females will lay up to 50 to 100 eggs in one egg sac that is covered in wax and protected in this fibrous material. The first instars are salmon colored, very active, very long legs, and not covered with wax. The second then they molt, like snakes do, and they'll molt into the second instar. It's at the second instar where we see a break between the female pathway and the male pathway. The female will continue to molt into 2 successive molts as becoming an adult female, changing slightly in body size and in coloration, while the male will develop a cocoon. And inside this waxy, fibrous cocoon will molt 2 more times as into the prepupa, and then the pupa form, and then eventually emerging as an adult male with its wings. And this development time is synchronized, and it's pretty remarkable to see that happen. And of course, once mating occurs, the female will then oviposit her eggs, and the cycle will, she will die after ovipositing those eggs. Okay, so in the vineyard, we can see what's called phenology. It's the grape mealybug life cycle through a seasonal analysis in the vineyards. And we have, thanks to Wendy, thanks to her for sharing this graph, we have data from March until August from 2 summers ago. And it's basically this is showing all the different stages that I've just introduced to you through scouting activities on the vines and being able to see when they are active, and then we can be informed on when we can spray and what we actually have in the vineyards. And we know that there's 2 generations a year. There's an overwintering stage, and then just before, just in June, July, is the next stage of mating. So it's a very informative graph, and we know that there's also, like I said, 2 generations. Okay, so monitoring for mealybugs, it's very important to know what you have in your vineyard and where they are on the vine. It involves peeling back the bark vine and actually using these pheromone-baited insect traps. Now, these pheromone traps are designed specifically from the sex pheromone of the female in order to attract the male. A very important tool for monitoring because it could identify hotspots in the vineyard and then relay information about the timing for treatments spraying. However, for the past 5 years, Wendy and her team have not been successful at catching as many males in the sticky card as we would expect. And this led us to really question why this was happening. And remember, pheromones are specific to a specific species. So we're speculating that perhaps the reason why the males are not being attracted to these traps is that we could be dealing with a different species or perhaps a subspecies of mealybug whose pheromone has not yet been developed yet. Which leads us to the mealybug taxonomy and understanding and identifying insects. The taxonomy of mealybugs is very tricky business. It involves making a microscope slide of these little small insects and then comparing morphological features such as type, shape, size, and pores of the insects in order to compare them and then eventually come out with their identity. But today, we mainly rely on molecular tools using DNA, and we compare sequences that are deposited on our global database, GeneBank. So for our project here, we sequenced individuals from one vineyard using the partial CO1 section of the genome, and then we compared this to other sequences from other vineyards that were collected in the Niagara region. So this phylogenetic, and we also introduced some genes from CO1 partial genes that were on GeneBank. Okay, and this phylogenetic tree was created by Dr. Cesar Pacheco, who is a mealybug taxonomist in Uruguay. And he started actually to work on this puzzle prior to my arrival to CCOVI. Wendy sent him the samples from 2 other vineyards in the Niagara. But this preliminary tree gives us also an indication that something is off with our identifications and our identity, because 98% of the insects that we collected had a 98% match to the insect that is called Grape Mealybug on GeneBank. So there's a 2% difference there that we'd like to further investigate with more robust genomic tools in the future to disentangle this mystery. But regardless of what its name is, the insect that we call the Grape Mealybug continues to be a pest and a vector of grapevine leaf roll virus, and therefore needs to be controlled. Controlling mealybug populations will reduce the spread of the virus. Roguing or the removal of infected material is a good practice to reduce the spread of virus. It's important to point out that there's no threshold for mealybugs. And so growers are, if one mealybug is found in a vineyard, a grower will then spray, regardless, just to try to eliminate the spread of the virus. A systemic insecticide, seen here, called Movento, is the most popular, commonly used insecticide against the mealybug. The active ingredient is a spirotetramat. This is a foliar spray that moves into the vascular tissues of the vine and is carried up and down the plant system to protect the leaf and the root tissue. So when it's applied to the foliage after bloom, it moves into the trunk where the mealybugs are feeding. And there's recommended times when to spray, but the fact that it's a systemic means that it targets the feeding behavior of the mealybug. So some other modes of treatment that have been approved by Canada and are presented here by the Crop Protection Hub are oils and soaps. They're less commonly used, and we also don't know, it's unknown toxicity for the oils on the honeybees. Therefore, there is a need and a growing demand for an alternative treatment for more environmentally friendly to bees, to beneficial insects, to growers, and biopesticides. This is where the entomopathogenic fungi they're now being considered in light of the rising demand for sustainable grapevine protection. So what are these entomopathogenic fungi? Entomo is insects and pathogenic disease causing. So they're basically fungus that are targeted for specific insects that will kill the insects. It's fungal spores that are directly penetrate through the exoskeleton of the insect and kills it. And here are some examples, pretty extreme examples of some fungus. Here we have a beetle, and down here we have an aphid. And there's different species. There's different species of fungus that will attack and kill different species of insects. So it can be also very species specific. Currently, grape growers are not using these products in Canada. And so we set out to see if this was an option for the grape mealybug. But in order to do some laboratory trials, we needed to establish a mealybug rearing in order to do that. We wanted to test two entomopathogenic, I will call it EPFs, throughout the remainder of the talk, against the grape mealybug, and not only which product out of the two and at what concentration would be the most effective. So in order to set out to set up a rearing in the laboratory, we needed to collect insects. Now, only once funding was approved, and that was September and October. This is when the mealybugs are starting to overwinter. We went out to one vineyard and peeled back, that was heavily infested. We peeled back the bark and with a paintbrush, collected the first instars into little vials that we then brought back to the laboratory. We also collected egg sacs that were on bark, and that we would bring back to the lab as well. So sorting through the material in the lab, we needed to make sure that we didn't have any hitchhikers like the ants that are associated with the mealybugs, as well as mites and other critters. We wanted to keep our rearing as clean as possible. We also cut pieces of bark that were infested and put them in vials so that we can store them in the refrigerator and then pull them out in a couple of months later for cold storage and to see if they actually would re-emerge. And spoiler, they did. So that was great that we had some insects on reserve. So the remaining mealies, the live insects, were sprinkled on potted plants, potted vines, and they're seen here. This method that we tried, we took infested bark and we propped it up against the vines, hoping that the mealybugs would move over to the fresh live vines, but they actually didn't. So sprinkling vines was the best way to go. We kept all the vines in incubator, and these were the conditions at 27 degrees. We only realized after the fact, after about 2 months, that we needed to do something about humidity in our incubator, and that's when we added a humidifier and tried to keep levels as high as possible. Okay. So I remind you that the size of the mealybugs that we were infesting were 0.5 centimeter. And so we needed some pretty powerful equipment here to be able to inspect on a regular basis. So my first major purchase, were these goggle lights that had 15 to 20 times magnification and had a light source. So it was able to detect them on the vines. However, during weekly inspections of the vines, Olivia started to notice that the mealybugs were dying. And there was a big question mark about why they were doing this. And so we took apart the vines. We thought maybe they were in the root system. But after consulting with the company that had given us the vines, we determined that they were actually treated with a broad spectrum insecticide just prior to infestation. So This was a very tough lesson for us, a difficult lesson, because we lost several hundred mealybugs that we had collected, but it's one of those, yes, this will stay in your mind, and the importance of using organic vines for rearing is definitely what would be recommended moving forward. We actually did have, interesting enough, we found male cocoons that were on these insecticidal vines. Now remember, males don't feed. So that was actually, we did get something from our rearing. We were able to take those males and move them to our other rearing, So not all was lost. And the other rearing included these vegetables. Now you're probably asking, why would we use vegetables for our vine mealybug? Well, using vines, potted vines, for research experiments and for a laboratory rearing, not the most efficient, especially when the insects hide under the bark. So we experimented with acorn squash, potatoes, pumpkins, and butternut squash. And we were very optimistic about the acorn squash because they actually molded into the second instar here. But suddenly, after 2 months, they died. So that was a loss. On the pumpkins, they actually never even molded. But the biggest success were the potatoes. And we know that I didn't just randomly choose these vegetables to grow these mealybugs on. We know from literature, from other scientists who have reared other species of mealybugs, that these are the more common, more successful fruit to use. But vegetables to use. And here we knew that we saw our first male cocoon. It's very small, the adult females, and then our first ovisac. So we were able to then determine that indeed potatoes could be the most viable and useful host for our rearing. And we've summarized here over the past half a year just the different host trials. And you can see obviously here in orange that potato was definitely the winner for being a favored food for the grape mealybug. So some rearing surprises and challenges. It's actually getting sprouted potatoes. You'd be surprised how long it takes to sprout potatoes when you actually need it. Rotting potatoes after a month, and then migrating females for ovipositing. Again, in nature, female mealybugs will move down to the bark, to the vine, for egg laying, and we saw some surprising things in our lab because of that. Slow development, dry eggs, and low hatching rates we also experienced. And until we perfected the humidity in our incubators, it remained an issue. So here I needed to show this slide because we spent a lot of energy trying to sprout our potatoes in the most efficient way with moist paper towel, with cloths under plastic, here even in soil. This was actually a surprise. It was a big lesson that we learned, that green sprouted potatoes will actually produce a toxin called solanum, and insects died when we put them on those potatoes. In dry conditions was actually the best bet. We had the highest sprouting, but actually it was a combination. It was a combination of first starting them out in moist conditions in light, and then about after a week, 2 weeks, putting them in dry conditions, and we were good to go. Our russet and Kennebec variety was also the more successful from the varieties that we tried. So rotting potatoes, like I mentioned, was an issue. We attempted to cut a potato in half, put it on a moist paper towel. That was one way of trying to preserve the potato for longer. Again, interestingly enough, once the potatoes started rotting, we were finding male cocoons in the rotted potatoes. Again, non-feeding males, so useful, but not quite. Removing sprouts from the potatoes, we also tried keeping them on moist surfaces. However, mealybugs were not happy with this situation, so we needed to abandon that fast. And thanks to Badra, we tried a leaf bioassay. But we know that mealybugs aren't really interested in certain leaves. But we needed to rule it out because we could have been surprised, and it would have been a lot easier for us to have this leaf bioassay on agar. And so what really actually, okay, sorry. So when I talked about the migrating females, this is a shot of the inside of an incubator, and thanks actually to Professor Glenn Tattersall from Biological Sciences. He lent us this incubator that we were able to use a third month in. We have a little humidifier set up here, and on the top shelf we have containers, plastic containers of the potatoes that were infested with the mealybugs, and a bucket of water at the bottom. Now, to our surprise, and lesson learned again, females would migrate from the potato onto the plastic container and then lay their eggs. Similarly here, eggs were laid on the cap of a vial, and the biggest surprise of all was when Olivia fished out an ovi sac that was in this container full of water at the bottom. Now, all these insects emerged as successful 100% emergence rates. So the lesson learned here is that female mealybugs do not need live material to lay their eggs on, and they also need very moist, wet conditions to do so, to have successful hatching rates. However, slow development, dry eggs, and hatching rates were main issues. You can see on this graph over a period of 8 weeks, that's 2 months. Now remember, we only had 8 months for this entire project. We see the blue are the instars, and the females only after the 3rd week started to be mature into females. And that means ovi sacs here was in the 7th week and 8th week. So we're talking about a 2-month life cycle, not something that I had planned for. Although when looking at its phenology and what it does in the vineyards, it's pretty similar in terms of its development time. I guess I was just hoping that in the laboratory, things would be a lot speedier for our experimental purposes. And I needed to show this picture because this was just in the last month that I invested in a chicken egg incubator in order to get those humidity levels up, and it's adapted for insect rearing. And yes, an insect rearing incubator is now on my wish list for equipment to move forward. So just some conclusions to wrap up from our rearing trials. We know we need 80% humidity to develop. The best varieties were russet and Kennick. Preserving the first instars in refrigeration is actually something that was useful and that we learned we can reinfest after that. So keeping the insects on, ice, so to speak, for when we need them. And that the females need bark in order to oviposit or some substrate. And so we've just added now bark to the containers for them to lay their eggs on when it comes time for that. But instead of having several thousands of insects, as I envisioned when I first started in September, and what we had planned for, there was only several hundred mealybugs that we ended up with. And so we had to tweak our experimental design slightly in order to accommodate for such low numbers. But nonetheless, we were able to continue with our original plan of doing the experiments using the entomopathogenic fungi. Now these 2 products were donated by Plant Products, and one is Botanigard and the other one is Lalgard. Both of these products are actually known to kill mealybugs, just not mealybugs in grapevines. So we already had a head start with knowing that they would be effective, but we were curious about obviously concentration rates and treatment plans, and that's what we set out to do with our several hundred mealybugs. So this is the experimental design that we set up. Each potato was infested with 5 second instars, and we had 2 concentrations for each product. So we had a low and a high concentration. And then we had the control here that we just used distilled water. And we did this experiment twice. And here you have Badra who's preparing the treatments, the saturated potato once it was sprayed with the product. After it was sprayed, the container was covered with cheesecloth and then placed in the incubator at the same conditions that we were keeping our rearing in separate containers. This one, each for separate container for each product, to avoid cross-contamination. And then, of course, daily observations for a 10-day period, looking for insect mortality. How am I doing with time?
You've used 35 minutes.
Thank you. Okay, so, and these are the results of this experiment. Now, this is what the insect started off as, a pink, soft, second instar, very, very small. And then after we sprayed, even on the second day, we noticed them looking shriveled, brown, dried out, dead. So we know that the product works. We actually, if you look here at the graph, here we have each day of observations and the percentage of mortality for the two experiments combined. Sorry, the average of the results of the two experiments. And what you can see is the higher concentrations had lower effect than the lower concentrations, and that mortality actually started from day 2. From day 1. But the highest mortality was 80% on day 7. Sorry, I think I have on day 7 here, and when we just have to decide which product would be better, it was a 10% difference between the Bavaria species and the Midorrhizium species. One was at 80% and the other was at 90%. So even though we had a small amount of insects, this graph and this experiment shows us some trends in terms of which products we can consider in the future. It's also we would definitely need to do more experiments with larger amounts of insects. And also different stages of insects. Originally I had planned to test the products on ovisacs and females, and we just tested on the second instars. Another interesting avenue for research would be isolating the fungus from the dead instars, just to have a confirmation that that is actually why they died. And Badra has gotten started on that, and maybe that will be our next CCOVI talk on the results from that. Obviously, these are lab trials, so whatever we do in the lab, it would need to be replicated in the vineyard, and there's multiple conditions in the vineyard to make this potentially a viable treatment. But we would like to do some vineyard trials as well with these products that are commercially available. Of course, the whole mystery around the genetics of our grape mealybug is something that we would like to further pursue in terms of developing pheromones, in terms of really isolating what mealybug we have here in Ontario and in Canada. And then, of course, other mealybug trials. There's other species of mealybugs, not potentially in grapes, we're not sure what variety we have of mealybugs, and then in the greenhouse applications for propagation, for virus, clean plant material. These are the avenues that we're considering for future research. So I would be remiss if I didn't acknowledge all the people, not only my partners who are part of the project, but the grape growers for allowing us to collect mealybugs from their vineyards, Casey Borowski from Plant Products for donating the products for the trials, Wes Weens of VineTech for donating the 20 potted vines, Rachel Bird and Quinn Nixon, who are summer students of Wendy. I call them the Grape IPM crew, who would be bringing me samples all the time during that summer period. Professor Glenn Tattersall for the incubator loan. Tony Wang, who's here for the CCOVI virus testing services. I was really surprised we were able to get some DNA from such small insects, but well done on you for doing that. Dr. Ian Scott, who also rescued me when I needed sprouted potatoes, gave me 5 kilos of Kennebec potatoes, already sprouted, as a gift back in December. He's an entomologist at Agri-Food Canada in London. And Dr. Fisher, who also brought me potted vines from Quebec out of desperation. You're not supposed to ask for potted vines in September. Now I know. And last but not least, Stephen Renda and his crew at Technical Services for helping to create my lab greenhouse here at Brock University. And to all of you for being here, for your attention here, and those people online, thanks for your attention. And I look forward to hearing any questions.
So do we have some questions from the audience?
Hi, Malkie.
Hi.
I'm just wondering about the potatoes and like mealybugs in the wild. Like, do they tend to infect potatoes in any way, or was this just kind of like a special lab treat that they tend to prefer?
Good question. So other species of mealybugs are known to feed on potatoes. This particular one, reluctantly so, from my experience.
And it was by far the preferred food source amongst all of the other like,
That's
small
correct. Yeah, and I'm sure it has to do with the sugar, carbohydrate, starch factor of the potato. Because they were excreting honeydew, they were developing. Very slow, mind you, but they were still doing what I set out to ask them to do. Yes. Potatoes, I know, it seems so random, right? A vine versus a potato, but it worked. Yes?
So I'm sure most of us know that if you see ants, they like to farm mealybugs in vineyards. Are there any other pests we could look out for that would be an on-off sign of like maybe there's mealybugs going on in there too?
Sorry, I didn't understand.
So like, you know, if you see ants, they like to farm mealybugs, and so if you see them, you can have a good sense there's probably mealybugs in your vineyard too. Are there any other insects we could look at in vineyards if we see them? It would be a good sign there's also probably a bunch of mealybugs in your vineyard as well.
Well, interestingly, because when we were, and it's a good question, and the whole question around ants is also a fascinating research subject that, I mean, I've collected those ants, I've preserved them in ethanol, I hope to identify them and really understand what they do and their involvement in moving mealybugs from tree to tree. So this is part of the epidemiology also of the virus transmission. That is a good question. But to answer your question, no, I do not know of any other insect that's associated with mealybugs right now that could be a sort of indicator that there are mealybugs. There are beneficials. I did find very many mealybugs that were actually parasitized. So we know that there is parasitism going on, and this could be a potential natural enemy, natural enemies to look at. The beneficials, the good ones. But yeah, good question. There were several, at least 3 different ant species that I found, that we found in the vineyard.
Malkie, I just want to commend you for your stick-to-itiveness with this. I know there were a lot of trials and tribulations trying to figure out the conditions to get the colonies reared. So well done on that front. And look, you know, you're on your way now with the research. So we're looking forward to hearing updates as this research program progresses. And, you know, some really interesting insights, you know, on these fungal pathogens that can control the mealybugs. So, great. On behalf of CCOVI, I'd like to give you a little gift for us, and we look forward to more talks in the future. So, thank you. So next week, just a reminder, please join us again, same time, same place, to hear Dr. Jen Kelly, who's a scientist in enology within CCOVI. And she's going to present on treatments to manage tannins in red wines and what the sensory impact are of those treatments on the wines after those treatments are completed. So we look forward to hearing from Jen next week, and we'll see you here 2 o'clock next Wednesday. Thanks.